The three important techniques of biotechnology. Hello everyone, welcome to Biotech World. Today let us understand about the three important techniques of biotechnology, which are recombinant DNA technology, means genetic engineering, plant tissue culture, and the transgenic, means genetically modified organisms. First of all, let us discuss about our first technique, that is recombinant DNA technology. Everybody knows about it, that all have studied about this in the 11th standard and the 12th standard. After this, it depends on the interest that a child goes towards which profession. Okay, let us start. Genetic engineering deals with synthesis of artificial gene, repair of gene, combining of DNA from two microorganisms by forming recombinant DNA and manipulating the artificial gene together with the recombinant DNA for the improvement of microbes, plants, animals, and human beings. Genetic engineering or the recombinant DNA technology involves cutting and pasting of desired DNA fragments. It is based on two important discoveries in bacteria. First one is the presence of plasmids in bacteria, which can undergo replication along with and independent of chromosomal DNA. Second is restriction endonucleases, which can break DNA at specific sites. They are appropriately called the molecular scissors. The science of recombinant technology took birth when Cohen and Boyer in 1973 were able to introduce a piece of gene containing foreign DNA into plasmid of Escherichia coli. It also discovered by the Berg in 1979 was able to introduce a gene of SL40 into a bacterium with the help of lambda phase. Berg is often considered as the father of genetic engineering. Let us discuss about some basic tools in recombinant DNA technology, which are enzymes. These include lysing enzymes, lysozyme, cleaving enzymes, also known as the enzymes. For example, exonucleases, endonucleases, restriction endonucleases, and the other is synthesizing enzyme, for example, reverse transcriptase, joining enzymes known as the ligases, and the alkaline phosphatase. Our second important is the vector or the vehicle DNA, the DNA used as a carrier for transferring a fragment of foreign DNA into a suitable host is called the vehicle DNA or the cloning vector or gene carrier, which can be the plasmid, bacteriophage DNA, capsid, an artificial plasmid, phagamid, phasmid, artificial chromosome vector. Our third one is the passenger DNA. It is the DNA which is transferred from one organism to another organism by combining it with the vehicle DNA. Complementary DNA, or also known as the cDNA, synthetic DNA is referred as the sDNA, and the random DNA. First, let us study about the complementary DNA. That it is the desired gene is prepared from mRNA by a reverse transcription process, and the product is complementary DNA, also known as the cDNA. Next is about the synthetic DNA. The desired gene is synthesized using a computerized machine called DNA synthesizer or DNA gene machine. This machine can synthesize gene of more than 1000 nucleotides length according to a predetermined sequence. If the number of desired genes is not enough for cloning, gene is amplified using polymerase chain reaction. Third is about the random DNA. The desired gene is isolated from the total genomic DNA of an organism. To isolate a gene, the genomic DNA is cut into a number of pieces with the help of restriction and nucleus. The pieces of DNA are separated on the basis of their length using electrophoresis and later on can be used for cloning. Artificial chromosomes from bacteria and yeast, called as the bacterial artificial chromosome and the yeast artificial chromosome, which are more efficient for eukaryotic gene transfers. Let us discuss in detail about the recombinant DNA technology. It involves several steps, which are: first one is the isolation of DNA segment. The DNA to be used as vehicle or passenger is taken out from the cell by lysing it with a suitable enzyme. The DNA is isolated from other cell content. Then both vehicle and the passenger are cleaved by using the same restriction endonuclease so that they have complementary sticky ends. Formation of recombinant DNA. The complementary sticky ends pair and their ends are sealed with ligase. This produces a recombinant DNA or the chimera DNA. Next is the production of multiple copies of recombinant DNA. Next step in this process is the production of multiple copies of recombinant DNA. Now let us study about the introduction of recombinant DNA into the host. E. coli, Bacillus subtilis, and the yeast are used as host for the recombinant DNA. Bacteria to be used as host should be without plasmids. Three methods are used for the introduction of recombinant DNA into the host. First is a transformation. It is the process by which a cell takes a black DNA segment from the environment, incorporates into it its own chromosomal DNA. Second is about the transduction. It refers to the transfer of DNA from one organism to another through a bacteriophage. Third is the vector-less gene transfer. Gene transfer can be effected by certain means that do not use vectors. It may be done by microinjection or gene gun. Next point is about the screening of transformed cells. The next step is to determine which cells harbor the recombinant DNA molecule containing the gene of interest. The desired clones can be selected on the basis of the presence of the vector of the inserted gene itself. For example, some plasmid vector confer resistance to an antibiotic. Another approach is to determine which cell bind RNA that is complementary to the gene of interest or synthesized protein encoded by it. Clones containing recombinant DNA are stable for at least several hundred generations. So guys, thank you so much. For further lecture, please subscribe and